Xellbiogene is a biotech company founded in 2013 by the Bambino Gesù Children's Hospital and the Catholic University of the Sacred Heart. In its laboratories, Xellbiogene will develop and produce biological drugs for clinical trials of advanced therapies and for consolidated oncohematologic therapies. Xellbiogene will work for the two founding hospitals, for research centers, pharmaceutical companies, other hospitals, and charities. The main goal of Xellbiogene is to contribute to the development of new therapies for currently incurable diseases. Xellbiogene operates in Rome in two of the largest production sites in Europe. The first one is located inside the Bambino Gesù Children's Hospital in St. Paul outside the walls, while the other one is located inside the Gemelli Polyclinic. The facility in St. Paul covers an area of 1,300 square meters, of which 500 are destined to drug production. In detail, the 500 square meter area includes 5 aseptic rooms for the production of cell therapy drugs, 3 aseptic rooms for the production of gene therapy drugs, 1 room for the production of viral vectors, 1 room for viral vector purification, 1 room for the procedures related to oncohematological transplants. Each of the rooms used to produce advanced therapy drugs is equipped with 4 220-liter incubators used for the concomitant production of biological drugs destined to several patients involved in the same trial. The production of drugs destined to clinical trials must be in compliance with the good manufacturing practices. The good manufacturing practices guarantee the quality and safety levels of medicinal products through the definition of the requirements regarding rooms, equipment, controls, product release, management of raw material and finished products. According to the good manufacturing practices, the process of cell and virus manipulation must be carried out in an aseptic environment in order to prevent antimicrobial contamination. Xellbiogene's laboratories include aseptic rooms exclusively used for the production of cell and gene therapies. Such rooms are equipped with an air filtering system that uses the HEPA 14 air filters, an environmental monitoring system to control and manage air filtering depending on the environmental conditions. In addition, the good manufacturing practices prescribe that all equipment and machinery must be periodically tested and measured. The rooms must be adequately cleaned. Operators must wear suitable clothes and be trained on how to work in sterile rooms. Xellbiogene's laboratories have been designed to adopt the most innovative technical solutions to assure compliance with the good manufacturing practices. The air filtering system consists in 14 units, each linked to one or more rooms. Therefore, in case of damage or maintenance, the production process can be suspended only inside a circumscribed area of the laboratories. Through the air filtration system, it is possible to set and control temperature, pressure, and humidity levels. Air filtering is provided by the HEPA filters, which retain 99.995% of 0.3 micron particles. All air conditioning system and equipment is monitored through the environmental monitoring system installed on the Siemens Desigo platform. In this way, any anomaly is detected and recorded so that, in case it exceeds the limit, the alarm sets off. The clean air leads into the sterile area through rooftop air filters and is subsequently sucked to prevent the airflow from rising, carrying dust or bacteria. Besides, the traditional suction grids have been replaced by air walls, which significantly improve the efficiency of the sucking process. Inside the sterile rooms, Each detail has been designed to prevent the formation of dust and bacteria. Some of these are closing the chimney of the biohazard hoods through a metal cutter that reaches the roof, steel tables with rounded corners, which bending radius equals the radius of wall corners, placing the fridges inside the wall so that the engine of the fridge that generates chaotic airflow is sealed off outside the sterile room. Color codes are used so that the different working areas can be easily recognized by operators who are trained to adopt appropriate area-specific procedures. Green refers to the floor of the rooms with controlled microbiologic contamination, while light blue identifies the floor of non-classified areas. Different door colors are used to identify different production areas. Yellow doors identify the rooms used to produce cell therapy drugs, while orange refers to the doors of the rooms used for gene therapy production. During the last decades, institutions, researchers, physicians, and biotech companies have multiplied their efforts in trying to develop reliable and adequate therapies for incurable diseases with a high social impact. There is still a long way to go that requires serious and constant commitment, so that, one day, clinical research will offer real therapies to the highest number of patients.